What a biosimilar could match its reference product almost atom for atom, same sequence, same fold, same glycan map, and still fail in approval. It happens not because the science is impossible, but because proving similarity is a discipline of its own, and a handful of avoidable mistakes derail program after program. Hello, I'm Raghuvar Singh Parmar, Director Scientific Affairs at ResolveMass Laboratories, and I work with biosimilar teams almost every day to build the analytical case that proves their molecule is highly similar to the reference. Today we are walking through the 5 critical mistakes that sink similarity claims and exactly how to avoid each one. Let's start with why this is hard. A generic small molecule is a single, well-defined chemical. Prove the same structure and you're essentially done. A biosimilar is a large protein grown in living cells, and no two batches are ever perfectly identical, not even the originators. So regulators don't ask for an identical copy, they ask for a totality of evidence. Analytical similarity at the foundation, then non-clinical, then a focused clinical confirmation. And here is the key: the stronger your analytical data, the smaller your clinical burden. That foundation is where the program is won or lost. So where do teams go wrong? After years of watching these programs, the failures cluster into 5 recurring mistakes. 1 is stopping at structure. 2. Leaning on a single reference slot. 3. Trusting one analytical method. 4. Treating every difference as equally important. And 5. Bolting the analytical strategy on late for a single market. Let's take them one at a time because each one is both common and completely preventable. Mistake 1 is stopping at the structure. It's tempting because the structural data is satisfying. Your sequence matches, your disulfide bonds match, your glycan map overlays beautifully, but that only proves the molecule looks the same. It does not prove it works the same. Structural similarity is necessary, but it is not sufficient. You have to demonstrate function, potency, receptor binding, and actual mechanism of action through well-designed cell-based and binding assays. 2 molecules can look identical on every chromatogram and still behave differently in a patient function is what closes that gap. Mistake 2 is relying on a single reference slot. Sponsors buy 1 or 2 batches of originator, compare against them, and call it similarity. The problem is that the reference product itself varies batch to batch and across its own life cycle of manufacturing changes. If you only sample one lot, you can't separate normal variability from the real difference. You might flag a difference that doesn't matter or miss one that does. The fix is to characterize many originator lots, map the reference product's true range, and judge your biosimilar against that range, not a single snapshot. Mistake number 3, trusting one analytical method. No single technique sees everything. Mass spectrometry is the gold standard for primary structure, but it won't fully resolve higher order folding, and folding is exactly where subtle safety relevant differences hide. The answer is orthogonality. Confirm every attribute with independent methods, peptide mapping and high resolution MS for sequence, circular dichromism and hydrogen deuterium exchange for structure, LC-MS, MS and capillary electrophoresis for glycans and modifications, size exclusion and light scattering for aggregation. One method gives you one blind spot, orthogonal methods give you confidence. Mistake number 4 is treating every difference as equally important. With this many orthogonal methods, you'll find differences. That's totally normal. The mistake is either panicking over all of them or worse, waving them away without justification. Not every analytical difference is clinically meaningful. The real skill is determining which ones are. So rank each equally attribute by its impact on potency, pharmacokinetics, and immunogenicity, and tie that criticality back to the mechanism of action. Then investigate your high impact differences thoroughly before a regulator asks the question you didn't answer. Mistake 5 is bolting the analytical strategy on late and for a single market. Analytical similarity isn't a box you tick at the end. It starts at candidate selection, runs through your comparative similarity assessment, and continues into bridging studies every time manufacturing changes. And it has to satisfy more than one regulator. The FDA and the EMA don't expect identical packages. At ResolveMass, we have supported sponsors who came to us late, mid-submission with differences they couldn't explain. And the fix often meant new studies and lost months. The teams that plan the analytical story early across the regions almost always move faster. So what should you walk away with today's session? Prove function, not just the structure. Compare your molecule to the reference full range, not a single lot. Use orthogonal methods and rank every difference by its impact on the patient, and build your analytical strategy early for every market you intend to enter. In the coming videos, we'll build the analytical similarity package itself, the one regulators accept the first time. If you are developing a biosimilar and you want a CRO partner who treats analytical similarity as the foundation of whole program from the first characterization to late submissions to the regulatory authorities, ResolveMass Laboratories supports biosimilar teams with mass spectrometry, functional assays, and regulatory ready data. Subscribe this video channel so that you don't miss the rest of informative series videos, and you can also drop a comment if you are working on biosimilar or this kind of very niche field. We would be happy to see these informative videos are working well. Thank you so much for watching this session, and see you in the next videos. Until then, goodbye for now.